Hello, everyone. Thank you so much to the OTS for honoring me with this award and inviting me to speak today about the RNAi modulation of APOE as a potential therapy for Alzheimer's disease. Alzheimer's disease is a progressively degenerative neurological condition characterized clinically by memory loss, dementia, and depression. Pathological hallmarks of the disease include hippocampal atrophy and postmortem presence of amyloid plaques and neurofibrillary tau tangles. While early onset Alzheimer's is caused by any mutation in amyloid precursor protein or its machinery, late onset Alzheimer's, which is the more common form of the disease, falls into a multi-hit paradigm where multiple genetic and environmental risk factors may interact to cause disease. One such risk factor involves the APOE gene. APOE is part of the larger family of lipoproteins. In the CNS, it is primarily expressed by astrocytes, and it functions in lipid transport between cells and the inflammatory response. Systemically, it is secreted by hepatocytes, and it is necessary for the uptake and clearance of chylomicrons and other lipids from the circulation. In humans, there are three allelic variants of APOE, E2, E3, and E4. The presence of one or two copies of APOE4 significantly increases the risk of developing late onset AD and decreases the age of clinical onset. Interestingly, when APOE is genetically removed in Alzheimer's disease mouse models, there is a reduction of Alzheimer's disease pathology, making APOE an interesting candidate for silencing with RNA interference. However, the removal of whole body APOE in mice also leads to the development of high cholesterol and atherosclerosis. Thus, in order to develop a potential therapeutic targeting APOE, we have to think about both brain and systemic APOE, both of their roles in neuropathology, and both of their roles in systemic cholesterol physiology, leading us to ask the question, what is the impact of each pool of APOE, CNS, and liver on neurodegeneration? Furthermore, is silencing of brain APOE sufficient to result in an improvement in neurodegenerative phenotypes without impacting systemic cholesterol homeostasis? And is it possible to silence CNS APOE with no effect on the systemic pool of APOE and no systemic exposure? Fortunately, the tools to answer these questions exist in the form of siRNAs. GalNAc conjugated siRNAs effectively silence expression in hepatocytes. And in the Corova lab, in efforts led primarily by Julia Alterman, Bruno Godinho, and Matt Hassler, we recently identified a novel chemical scaffold called the diSiRNA that distributes throughout the entire brain of mice and non-human primates after a single ICV injection and causes potent target silencing for at least 6 months in mice and 1 month in non-human primates. Using these tools, we began to investigate our questions. We performed parallel studies in the APP presenilin-1 mouse model of Alzheimer's disease. In one group, we used the diSiRNA targeting APOE to silence CNS APOE, and in another group, we injected animals with GalNAc conjugated siRNAs to silence hepatic APOE. We treated all animals at 8 weeks old prior to the onset of symptoms and neuropathology, and we assessed all animals at 16 weeks old after documented neuropathology forms. We first evaluated animals for mRNA and protein expression after treatment with the diSiRNA. And as you can see here in the hippocampus and cortex, we have potent knockdown of APOE mRNA and APOE protein 2 months post-injection. We do see some silencing of liver APOE, approximately 50% at this time point, where we believe the diSiRNA to be cleared. In our second group that we injected with GalNAc conjugated siRNAs, we have potent silencing of hepatic APOE mRNA and protein 2 months post-injection with no impact on brain APOE expression. Next, we evaluated the impact of silencing APOE on amyloid plaques. Using immunofluorescence and X34 staining as orthogonal assays, we can see amyloid plaques in our control groups. In our treated groups, we were excited to observe a decrease in amyloid pathology, which was significant in both male and female mice, but with a more pronounced difference in female mice. Amyloid burden can also be measured using ELISAs for A-beta 40 and A-beta 42 fragments. Consistent with plaque data, we observed a reduction in both A-beta 40 and A-beta 42, with the most significant reduction being in A-beta 42, the more toxic fragment in female mice. To further understand how silencing APOE may improve phenotypes, we performed RNA sequencing on a subset of brain samples. First, we can see in the volcano plot that of all the differentially expressed genes, of which there aren't many between treated and control groups, APOE is the most significantly downregulated. We observed 22 other differentially expressed genes and 3 pathways with significant gene enrichment, including fatty acid homeostasis, positive regulation of peptide secretion, and regulation of peptide secretion, identifying areas for future study. In our parallel experiment where we silenced hepatic APOE, we did not observe any differences in amyloid plaque burden or A-beta 42 fragments at the time points tested, suggesting that hepatic APOE is not contributing to pathology at this time point. Importantly, after the removal of brain APOE, we did not see any increases in serum cholesterol. In comparison, after removal of hepatic APOE, we saw a significant increase in LDL cholesterol, suggesting that silencing brain APOE, at least at this time point, is sufficient to improve amyloid phenotypes while not impacting systemic cholesterol homeostasis. Next, we sought to devise methods to silence only CNS APOE and prevent any hepatic silencing from systemic exposure. In wild-type mice, we once again injected the siRNA targeting APOE into the lateral ventricles, and at 1 month post-injection, we were excited to see potent mRNA and protein silencing throughout all regions of the brain. And once again, we observe approximately 50% silencing of APOE mRNA and protein in the liver. We first tried to mitigate the systemic exposure by reducing the dose. And as you can see here, when we reduce the dose in 2 and by 4, there is a rescue of liver silencing and brain silencing is maintained. But we wanted to see if we could devise a way that we could maintain high doses in the CNS and block liver silencing completely. In 2018, Zlatov et al. Published this beautiful paper using GalNAc conjugated antagomer-like reversiers to reverse the effects of siRNAs targeting TTR in the liver. We sought to build upon this approach and use a similar one to achieve brain-selective silencing. We designed 2 anti-siRNAs of different lengths that were complementary to the APOE targeting siRNA. We injected mice with a high dose of siRNA into the lateral ventricles and first tested if the anti-siRNA could rescue expression after silencing had already been achieved, and injected the anti-siRNA subcutaneously 1 week post-ICV injection. Second, we tested the blocking capacity of the anti-siRNAs by administering a pre-dose to the animals prior to their ICV injection. Because APOE is a secreted protein that is abundant in the serum and primarily made by hepatocytes, we were able to track expression over time by taking blood draws and running ELISAs that correlate back to liver silencing. After ICV injection of the siRNA, we collected blood each day, and you can see here that the amount of plasma APOE protein rapidly drops in response to systemic exposure in all groups. After subcutaneous injection of the 8-mer anti-siRNA, once silencing reached a steady trough, we observed a rapid recovery of systemic APOE protein, seen here in purple, compared to our PBS control in green. Similarly, after injection of the 15-mer, we see a rapid recovery of systemic APOE, although less statistically significant, which is consistent with the results in the literature. In our prevention model, we were able to completely block the effect of systemic exposure in both the 8-mer and 15-mer groups, as seen here, compared to our PBS controls in green. And once again, the 8-mer is more statistically significant. Most importantly, when we look at silencing in the brain 1 month post-ICV injection, we see sustained CNS silencing in all groups. And when we evaluate silencing in the liver compared to our PBS control, we see a rescue and a block of hepatic APOE expression. In summary, I've shown you today that the removal of brain APOE with the di-siRNA is sufficient to improve neurological phenotypes of Alzheimer's disease, that brain APOE is not necessary for the maintenance of cholesterol homeostasis. We've started to identify pathways and targets affected after silencing of APOE and hope to someday elucidate the mechanism of how APOE is impacting neurodegeneration. And finally, we've been able to achieve CNS-specific silencing through both dose reduction and chemical engineering. We're excited about these results and we're excited to pursue them further. And what we'd like to do is treat animals after the onset of pathology and see if we can reduce the worsening of disease and maybe reverse some plaque formation. And we'd also like to evaluate neuropathology at later time points. We'd also like to evaluate how the transcriptome changes at multiple time points and see how worsening disease impacts which genes are expressed. And we'd like to use different siRNAs targeting APOE to differentiate between changes in gene expression due to mechanism versus due to potential off-target effects. So with that, I'd like to acknowledge everyone in the Kurova Lab and at UMass Medical School for contributing to this work. We're a very collaborative group, and it's been a pleasure and a joy to work with everyone in the lab. Thank you very much.